Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 6th October 2020. As we are into the mood of revision, and in this chapter, principles of inheritance and variation, yesterday in our last session we were discussing about what is called incomplete dominance, mirabilis jalapa. Okay, and what we did, if you recollect from that, is we crossed two flowers. One is white colored and another one is red colored, and the resultant F1 generation is pink flowers. And this is possible because the incomplete dominance of the dominant gene, capital R. Now today we are going to talk about one more thing, codominance. Codominance means two genes, two alleles will share their influence, so we say codominance. It is the phenomena of two alleles lacking dominance, recessive relationship, and both expressing themselves. This is the thing. There is nothing called dominant gene or recessive gene here. Both are equally sharing their expression. So in others, A, B, O blood grouping are controlled by gene I gene. The gene has three alleles, as all of us know: IA, IB, and I0. I'll say IO or I0. Any person contains any two of the three alleles, like IA, IB are dominant over I0. I'll say IO or I0. The plasma membrane of the RBC has sugar polymers that protrude from its surface, and the kind of sugar is controlled by a gene. When IA and IB are present together, both express their own types of sugars because of codominance. So when there is presence of IA, IB, both will express. Hence, you get AB, blood group AB. So what is the basic difference between incomplete dominance and codominance? Effect of one of the two alleles is more conspicuous in incomplete dominance. Effect of both the alleles are equally conspicuous. In incomplete, it produces mixture of the expression of two alleles, like white flower and red flower. Mixture is pink. There is no mixing of the effect of the two alleles. There is nothing called A plus B by 2, that blood group. Okay, it's just AB. A is also expressing, B is also expressing equally, so you are getting AB blood group. The F1 does not resemble either of the parents. Yeah, parents we know white and red, and F1 is pink. F1 resembles both the parents. That is the thing. A, B, it has got antigen of A as well as antigen of B. So both the parents it resembles. Now flower color in dog flower, and here ABO blood grouping in human beings. Okay, and then we move ahead with another important concept called multiple alleles. There are multiple forms of Mendelian factors. Okay, N is missing. Mendelian factor or gene, which occur on the same gene locus distributed in different organisms in the gene pool, with an organism carrying only two alleles and the gamete only one allele. ABO blood grouping also provides a good example of multiple alleles. As we said, the multiple alleles are IA, IB, okay, and I0. So here the effect will be like IA, I0, for example. The dominant one is A, so blood group A. Similarly, IB, I0, dominant is B, so the person gets blood group B. And in case this IA, IB, both are there, the person gets AB blood group. And only when person has got I0, no crystallization on the RBC, then it's a O group. And of course, in addition to this, you must be hearing somebody A positive, A negative, B positive, B negative, AB positive, like that. This positive is the extra antigen that is present, that's the serum. Okay, so if that is there, then we say A positive, one protein is extra on that. Okay, If it is not there, all will be negative. A negative, B negative, AB negative, O negative, like that. And all of you know that the very rarest group is A negative. Right? Moving ahead, we were talking about until then inheritance of one gene, and related to that one gene, codominance, incomplete dominance, and multiple alleles. Now we are going to go with the two genes, inheritance of two genes. We call this as means two, dihybrid cross. A cross made to study simultaneous inheritance of two pairs of Mendelian genes. Okay, and this asserts that law of independent assortment. The law states that when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of their other pair of characters. Like here we are going to take round yellow. That is, round is nature of the seed, and opposite to this is wrinkled, and another one is color of the seed, yellow color and green color. So we are taking two characters. So when the law of segregation and law of independent assortment particularly say that when two pairs of traits are combined in a hybrid, segregation of one pair of character, what is that one pair of character, like round color of the seed, is independent of the other pair of the characters, that is wrinkled or this nature of the seed. So in dihybrid cross, two new combinations, round green, normally what you observe in the nature is wrinkled green, and wrinkled yellow. Wrinkled yellow is rare to see, round yellow is dominant, are formed due to independent assortment of traits for seed shape, round, wrinkled, and seed color, yellow, green. So keeping that in our mind, what exactly we do? Again, Mendel has performed this in Pisum sativum, garden pea, and he has taken two characters. What is the character? First, shape of the seed, round seed, you must have seen, very dominant. And how do you decide whether round seed is dominant or wrinkled seed is dominant? And the very common sense is that numbers. You just take a box of seeds, okay, pea seeds, and you count. You need not even count, just by observation you come to know that round nature seeds are more. So whichever character has got maximum number, that goes without saying that it is the dominant character. So round is a dominant character over wrinkled. Similarly, you must have seen seeds, pea seeds, yellow color is dominant over green. That's why round yellow is dominant. Once it is dominant, you have to write capital letters. Round, capital R, homozygous condition. Capital Y, again homozygous, means pure round, pure yellow. And this is crossed with wrinkled and green. Now here wrinkled, you need to take small, that is small r. Remember I told don't take WW because spelling starts with wrinkled. Whatever you are taking here, corresponding opposite letters only you must take. And similarly YY, capital yellow, don't write GG, green. No, corresponding one, small y. Now when law of segregation takes place in the gametes, what happens? The one allele, capital R, one allele Y comes from this parent, and in another gamete, one small r, one small y comes from this parent. So when gametes fuse together, zygote form, and the zygote will be having capital R from this parent, small r from this parent. So capital R, small r, capital Y from this gamete, small y from this gamete, capital Y, capital Y. So R, capital R, small r, capital Y, small y, that is the genotype. And this genotype decides the phenotype. Because of the presence of law of dominance, capital R, it becomes round. Because of the presence of capital Y, law of dominance, it becomes yellow. So the phenotype of F1 generation always is round and yellow. And when this is selfed, F1 generation species are going for self-pollination, then you get what is that? Capital R, capital Y, small y into same, because selfing. Capital R, capital Y, small y. So as per the Punnett square, when you do this Punnett square method, what do you get? You get the gametes from this capital R, capital Y. This is one combination. Small r, capital Y, another combination. Small r, small y, and small r, small y. Small r, capital Y, small r, small y. So you write all this 4. You can see here. Okay, 1, 2, 3, 4. So capital R, capital Y, small r, capital Y, small r, capital Y, and small r, small y. And a repetition of the same thing. So capital R, capital Y, first one, capital R, small y, small r, capital Y, small r, small y. So 4, 4. So 4 into 4, 16 products, zygote should be there. 4 gametes and 4 gametes. So 4 female gametes, 4 male gametes. So fusion of this. When they fuse together, what happens? Now we are going for this capital R, capital Y, and this gamete fuses with these 4 male gamete. So capital R, capital Y versus capital R, capital Y, you get all the dominants. Capital R, capital R, capital Y like that. And when this combines with this capital R, capital Y combines with small r, capital Y, what happens? So you get capital R, small r, and capital Y, small y. Still it is round and yellow. Next, capital R, capital Y combines with capital R, small y. Then you get capital R, capital Y, small y. Still law of dominance, round and yellow. And last, the capital R, capital Y versus small r, small y. Capital R, small r, capital Y, small y. Still you get this. So like this, when you take a paper, write all this, and when you start writing it, this male gamete versus female gametes of all the 4, you write in the checkerboard. Then this you make this gamete to fuse with all the 4, write the genotypes. Similarly, take this, make a combination with all the 4 gametes. Take this small r, small y, make the combination with this male gametes, then you get the zygote. Now, first, foremost thing, what you must do is write genotype. So, based on genotype, write the phenotype. And remember, law of dominance, don't forget that. So, at the end of it, at the end of the cross, what do you get? You get 9 round yellow, 3 round green, 3 wrinkled yellow, and 1 wrinkled green. So 9:3:3:1. Again, this is universal. Majority of the cases, unless there is some deviation, this is the principle. That is how we must appreciate Mendel. The ratio of 9:3:3:1 can be derived as a combination series of 3 yellow, 1 green with 3 round, 1 wrinkled. The derivation can be written as follows: 3 round, 1 wrinkled, 3 yellow, 1 green is equal to 9 round yellow, 3 wrinkled yellow, 3 round green, 1 wrinkled green. Of course, once you put this Punnett square, you will come to know how to do it. So what you have to do is, take a paper, write this, put a checkerboard, okay, or Punnett square method, and do it. And after doing this, as I said previously also, you take another combination. Like you take color of the flower, you take. So write genotype, color of the flower, and position of the flower, so that you can do yourself and classify, verify whether you are getting 9:3:3:1. Then only I understand that you understood the concept called dihybrid cross correctly, and post your answers in our group. Until then,